County Weed District, MOA is pleased to welcome John Gaskin with the USDA Northern Plains Ag Research Lab in Sidney, Montana, Mark Schwartzlander with the University of Idaho, Harriet Hines with the CABI Switzerland Center, and Melissa Maggio with the Montana Biocontrol Project. This session will be monitored by Bryce Christians, Missoula County Weed District Manager. A recording of this event will be posted on MOA's YouTube channel. If you would please put your name and affiliation in the chat, that would be helpful. That will help us give a record of who is attending. Also, I will help Bryce monitor any questions you may have in the chat. Please be sure to mute your microphone if you are not speaking. And now I'll pass it over to Bryce.
Thanks, Jamie. Yeah, I also wanted to take the opportunity to just thank everyone for joining us this morning. Harriet for joining us this evening, since you're joining us from Switzerland. I am really excited to have these speakers here today to have this conversation with you all. Missoula County Weed District's mission, or partly its mission, is to really build a connection between researchers and producers on the ground. So this is a great opportunity for us to continue to build that relationship. And I wanted to thank Jamie specifically for helping us organize this this morning. So yeah, Jamie introduced, we have 4 speakers today. And I think I'll just give a brief introduction to reiterate who they are and the organization that they work for, and let them provide their presentation. I think we'll leave a little bit of time at the end of each presentation for just a couple questions. And again, if you want to put your question in the chat, maybe we can pick up 1 or 2 questions before we move on to the next speaker. And then all of the speakers have agreed to stay on a little at the end if there's additional discussion or questions that we don't get to right after their presentation. So not to stall us any longer and to get into the information that we're really all here for this morning, I'd like to introduce John Gaskin with USDA ARS out in Sidney, Montana, and let him take it away.
All right, thanks, Bryce. Let me share my screen. Here we go. Can you see that all right?
Yes.
Yes.
And you can hear me? Okay. Yeah, I'm John Gaskin. I'm a botanist out here in Sidney, Montana. And one of my goals, my work, is to sort of spread the word about biocontrol development for field bindweed, and to make sure that the whole research process continues on, you know, looking for funding being part of one of those things right there. But I'm just going to talk for 10 minutes about field bindweed biocontrol. And this is work from Jeff Littlefield at MSU, who did a lot of the earlier stuff.
Trying to get it to it.
Ghislaine Cortat's actually doing the hard work in Switzerland, and Harriet Hines, who runs the Biocontrol Development Center there in Switzerland. And working on changing my slides. There's bindweed in all its glory. Maybe one of the top 10 weeds in the world, actually, and a big problem for, especially for organic systems, as you know. I don't have to tell you too much about it, but there it is. It's native to Europe. And one of the interesting things is it's on the left, you can see a picture. That's a distribution out of one of the databases, and you can see that it goes down to Puebla, Mexico, is about as far south. When it starts to get really tropical, it drops off. And on the right, you can see an EDMAPS distribution, and it's up there in Alaska, interestingly. So this weed can go a lot of places. On the right, far bottom right, you can see that USDA plant says it's in pretty much every state and most of the provinces of North America there. So what's my goal? Yeah, like I said, my goal is to make sure that the development moves along at a reasonable pace. That means a lot of communication, making sure there's adequate funding, and that the development and side research goes on. So first, I'm going to talk about the status of what biocontrol agents are out there for this. And it's not great news, but this is info from Jeff Littlefield. There is a mite, you can see a picture of it up on the left, Asaria. It's an areophid mite. It's from northern Greece, has a lot of generations, dispersed in the wind. It's in my yard, and it induces some galls on leaves. You can see on the far right what it does. You may have seen this because the mite may be around your area if you have a lot of bindweed, and it can stunt the plant. It was first distributed, this one was from long ago, decades ago when this one first came out, and distributed at the stars on the map. And you can see they say it's there in Fergus County and down near Billings. I know it's in other places too. It flies around in the wind, and it's gotten a lot of places. Big problem is it hasn't done real great in Montana, especially. In southern climates down toward Texas and stuff, yeah. So I don't think anyone's got a definitive answer of why it might be an ecological problem with moisture, things like that. But in Montana, it was widespread for a while, pretty abundant, but it's become less abundant in a 9-year study. So it's not helping out too much. There was also another agent developed long ago. It was Taita, which is a defoliating moth, just likes to eat on the leaves there. Several generations a year. It's had a limited establishment on that project. It was probably put in Montana in 1998, and it's established in a few places, but no one really relies on it to do too much. And it's out of Italy. So it's got little apparent impact, which is something that bothers us in biocontrol when we go to the hard work of developing something, bringing it overseas. And so that one's not the answer for helping organic systems in Montana, especially. But the good news is that I really want to talk about is what CABI is doing right now with 2 additional potential biocontrol agents for bindweed in North America. The promising ones here, I didn't know that's going to do animated, but I'll get it. It's the first one I want to talk about is the fly in the middle picture. We're looking for insects that'll target the roots. I mean, you can hammer on bindweed on the top above ground a lot, and it's got a lot of root mass, as you know, so that may not slow it down too much. So they are looking at this Melanogrammiza, which is the fly, and a Microphysia, which is a clear-wing moth on the bottom right now. Let me tell you a little more about those. First of all, when we do testing for biocontrol, I don't do it, but when they do testing for biocontrol, we have to make sure it doesn't attack anything native here in North America or any of, say, the important crops, especially nothing T&E, threatened and endangered. So bindweed's in the Convolvulaceae family. It's got a lot of species in North America, especially if you include Mexico, which has a lot of species down there, including, Ipomaeas, Evolvulus on the bottom right, some ornamental bindweeds on the left bottom, and of course, sweet potatoes are in there too. So lots of things to test before we get any clearance here. So what Ghislaine does is she's setting up tests, like here's the test for the fly, where she'll put one of our native species, takes it to Switzerland, grows it, and puts it next to bindweed that's in a field there, and then releases the biocontrol agent to see if it attacks the plant that we value. Sorry about the noise of the cat in the background. And with the fly, what she found in the field is that the fly is being very good. It's certainly on the top line. It's attacking field bindweed, which means it's either feeding on it, and then they look, is it laying eggs with larval development? And it's certainly doing that, but it's also doing it a little bit to Calistegia longipes, which is a picture on the right. That's a native in California, sort of on the edge of the Sierras, right there. And it did have some larval development. When it just attacking it, they don't worry so much because that might just be spillover, but larval development's a bit of a problem. But what she has told us is that it was the 2 plants, our native Calistegia longipes and the field bindweed were growing intertwined, because you know how bindweed is, it just starts taking off and spreading. So that might have confused the fly and the larvae right there. So more tests are going to go on with that, make sure it's good. But except for that, the fly appears to have very narrow host range under natural conditions. That's out in a field, a cornfield, I think it was, in Switzerland. As for the clear-wing moth, It's also native over there to Europe, Middle East, Central Asia. The larvae of this one, which is really excellent, feeds on roots, and it can migrate between the roots underground when the food's exhausted. And there is some, the larvae are overwintering in the roots too. So she's doing testing on that. The nice thing is these moths can kill several smaller plants, which is pretty good. So she takes eggs from these, Ghislaine does. This does not seem like easy work. And she puts them on a leaf like they are. You can see on my blue arrow right there. And the plants are kept growing, and they see if the eggs develop and the larvae attack the native plant. And this is really great in terms of biocontrol research results right here. You can see that the larvae are hatching on a lot of the field bindweed, and then a bunch, 42% and 58% of the plants have either larvae or mines on the weed. That's great. And none of the other native species are touched. That's fantastic when it comes to biocontrol. So what's the summary on this? The results of the host range tests for both species are really promising. And I think Ghislaine's gonna find us the information about the one that did get attacked with development, you know, as she'll grow them separate and make sure that the fly larvae aren't bothering the native plants. The observations suggest that the larvae of both insects can have high impact on the target weed. And this is impact in the top of the plant, going into the root crown and down into the root, especially for the clearwing moth, which is really excellent because field bindweed has so much root energy that we have to hammer. The fly is expected to establish at disturbed sites like crop fields. That's where it likes to hang out. So that's good for us. And the clearwing moth, it does prefer loamy soils, so maybe that's good for vineyards and orchards. Also could be useful for organic systems in the West. So Ghislaine's going to continue to proceed with testing these two potential agents, which are pretty exciting. And they're going to do some rearing colonies of clearwing moths, because some of these are a little hard to rear, but they're going to crack that nut too. So how do these projects get funded? This one has been, the fly and the moth have been funded mostly by USDA APHIS, which is another branch of USDA from us. They worry about pests in the USA, and they've supported the project for quite a bit, but for 35 to 40,000. But most of these biocontrol projects, ones you'll hear about later from others, are funded at the range of 50 to 80K per year. That's what it takes. And I will remind everyone, CABI is a nonprofit entity that works around the world, especially in a lot of poorer countries, helping with agriculture issues. So this isn't like they're making a big killing on money on this. They're truly trying to help the world with agriculture improvements. There's the budget, and it's mostly to pay for Ghislaine and a technician and some field trips and some indirect costs of running the facilities, which Harriet will tell you about a little bit. Their ideal budget's a little higher, maybe 25,000 is what they would like to have to keep this project rolling along well, because it takes a few years to do all this work. So they're missing about 25 to 30,000. My unit here in Sydney is putting some money toward it this year, but they're actively looking for other money. That's about it for what I want to say on field bindweed. I did want to mention that, you know, you all might be a little bit worried, especially in the small grains world, about ventonata and cheatgrass. And that's research going on with USDA-ARS in France and the BBCA in Italy. They're partners of us and a lot of groups too that are looking into those. So I just wanted to say that because I know that's not in CABI's purview, and Harriet's not going to mention that. So if you have any questions about field bindweed, if you want to talk about it, questions about it, about the research, about the science, about the biocontrol, I would love to talk with you. I'm the consortium lead for North America on it. And And then Harriet, who you'll hear from in a minute, is happy to talk with you, as well as Ghislaine, who is in Switzerland doing the hard work. And Jeff Littlefield down at MSU, who's going to retire soon. So if we want to get any knowledge out of him, we should do that soon. But I did want to say, if eventually, if anyone wants to talk to us about some funding, talk to me. I can't take money as a government employee, but I know how to in places. So with that, I'm going to stop sharing my screen right now. Thanks, Jamie.
Thanks, John. So there was one question in the chat. I think Mark addressed that. Jess, was that what your question was? Or if you want some additional clarification?
On the line, yes, please.
I'll let Mark do that.
Okay. Mark, are you there?
Yes, I am. Can you hear me?
Yep.
I hope that is what you were referring to, but in entomology and bug sciences, when herbivore insects feed inside a leaf, or the larvae feed inside the leaf, or feed inside a root, or feed inside a stem, we call them miners, root miners, or leaf miners, or stem miners.
All right. Any other questions for John on those two agents, or on ARS in general? And I can't see everybody's screen, so if you do have a question, I'm not calling on you, can raise your hand or just put it in the chat. I can find you that way. All right, if there aren't any other questions on this in particular right now, it doesn't mean if you have a question for John later, you can't put it in the chat and we'll catch it at the end of the session. But we'll move on to Mark Schwartzlander, who is a biocontrol researcher out of Moscow, Idaho, at the University of Idaho. And I'll let you take it away, Mark. Oop, and you're on mute.
Thanks, Bryce. And
Yeah.
I will also try to share my screen with you. And hopefully you see that. So thanks for having me. I take 5 minutes of your time to talk about the weed that has haunted my career. I worked on it as a graduate student, and 30 years later, I'm still working on it. And this is a talk by me and my lab, but I should also mention the CABI folks and Harriet, because they are instrumental and involved in rearing these insects and working on the petitions. So, biological control of houndstong. Let's see whether my screen, I get my screen to forward. No, stop share. Why is my screen not working? Oh, here it is. Voila. So houndstong is probably not a weed that all of you are interested in. It's a rangeland weed. It's very toxic because it has chemicals in it, pyrrolizine alkaloids, they're called, that are really toxic to mammals, and horses or other livestock can actually get really sick or die when they ingest the foliage fresh or dried. It has been introduced some 150 years ago as a contaminant of grains from Central Europe, and is known for its sticky seeds that are like Velcro. You see it, this little rust electron microscope picture where the little hooks are shown on the seeds, and it sticks to dogs and to cattle fur, which is the way how it is distributed. So for those of you who are ranchers or cattlemen, or who are into organic beef production, you will probably be quite familiar with this weed. And those of you who have dogs and go hiking a lot, you might have encountered it too. So John mentioned that nicely, and I don't have time to go into this whole host specificity testing, but when we try to get these biocontrol agents here, what we have to ensure is that they do not attack native plant species, and certainly not those that are protected or threatened and endangered listed by the Fish and Wildlife Service. And in the family in which houndstong is, the Boraginaceae, there's quite a number of them. Well, 5 actually. You see those here in the picture. And also some close related North American species that are used as perennial ornamentals. Delinia grande is really nice flowering plant that is sold and maintained at the West Coast in many areas. So here's the fifth that an endangered species. It only grows in Texas, but I show you this picture because this is the species that stopped us from introducing the first biocontrol agent for hound's tongue in 1997, and that is already a long time ago. And I explain why when I get to this insect. So I'm talking about two insects in a minute. Oh, here you see the distribution of these threatened and endangered species in the country. Typically, all these species only have one or few populations, and they only occur in one area of the country. These are county levels, but actually they are much smaller than within the county. The one you see in the south in Texas, it's really 9 population within an area of 5 square miles, just outside Big Bear National Park to the north of it. But we just have to make sure that our insects do not pose any threat to these plant species, which is why we do a lot of host specificity testing, which is why John has talked about, I forgot totally what he talked about, the weed that he talked about. Because field bindweed and ventinata, because these host specificity testing or host range testing is quite expensive and it takes a lot of time. So there's 2 biocontrol agents I talked to you about for hound's tongue: a seed-feeding weevil and a root-feeding weevil. The seed-feeding weevil we studied since 1993. It has quite a bit of impact because the larvae mine in the seeds, so they destroy the developing seeds, and the adults feed on the flowers. And combined, they really reduce those velcro seeds by quite a bit. And after a lot of testing, we submitted a petition last September, and we got 3 weeks ago a letter from technical advisory group that basically recommends to USDA APHIS. John mentioned that agency, and that is the agency responsible for releasing these insects or permitting the release, that they recommend the release. And with that being said, we really hope that next spring, not this spring, we have the official permit in hand to release these insects. And you see on the lower right there, a graduate student of mine, Angela Tapper, who's currently rearing the insect and beefing up the rearing, and she's also doing some other things, of course. So spring 22, we hope to get these guys out. What we did for the weevil is, in addition to what the folks at CABI do and what John talked about, we did a lot of chemical ecology, where you basically study the response of these little weevils to the scent and the colors of flowers of the host plant and other plants. And it is really great data because it helps you to explain when an insect might nibble on one of the plants that we know it's not its host plant. So we get really great data out of it, but it also takes quite a bit of time and lab work and poor graduate students who have to slave on this kind of work or research. So then there's Mogulonis crucifer, the hound's tongue root weevil. We have petitioned that weevil in 1993 and in 1996, and both times APHIS rejected or denied the release of the weevil in the United States because it has the broad physiological host range, meaning that if you force the insect onto a plant, it will feed on plant species, on some native plant species, even on some TNE plant species, and the Fish and Wildlife Service didn't like that. In Canada, where they have much less non-target species or plant species in the Boraginaceae family, they did release the weevil in 1997, and it's tremendously successful up there. So while we are not allowed to move the insect here, it has crossed the border in 2008, and now it is pretty abundant in northern Idaho, northern Washington, and northern Montana. So again, what we try to do then is we try to use this chemical ecology to see whether we cannot really demonstrate to the federal agencies that this insect actually is safe. And I don't go through this, but this is the kind of data that you get and can publish. This is talking about arrestment. So when a herbivore insect that is specific to a plant species find its host plant, it gets literally arrested. It doesn't see a reason to move any further because its food source has been identified. And then what you find is that on non-target plant species, it doesn't do that. It continues to move on. And this is what this graph shows on the right side of these graphs. Now we have developed a really sophisticated, complicated test design that we think will allow us to convince even the greatest skeptics that this insect actually does not attack any non-target plant species, let alone the threatened endangered plant species, should it be released in the country. And this is a two-way system. Once where we, on the one side, we do experiments in the laboratory, and then we try to verify them in these large field cages that are 24 by 36 feet. So we have done that for quite a number of plant species. Last year, we hoped to get more than 4 done this year. And while we have a longer list of plant species that we need to accomplish or want to accomplish, meanwhile, we write a petition for this weevil anyways, because we have collected a lot of this chemical ecology data, and we think it's pretty strong at this point. The petition, as it's called, for release should go in the spring. And we're cautiously optimistic based on what the federal agencies responded or replied to us with regard to the seed feeding weevil. And while we let the guys in Washington, D.C. decide on that petition, we will continue to collect data on that Houndstone root weevil. Ultimately, we hope to release both. And if you have any questions, there's my email, there's my phone number. Thanks so much for listening to me.
Thanks, Mark. I don't see any questions in the chat, but curious if there are any quick questions for Mark before we move on to Harriet. Ole.
I was wondering if you could explain chemical ecology in a little more detail. If I understand you correct, it's when the insect bites on the plant, then there's a reaction in the plant and you can measure that. Is that correct?
That you could do too. And a lot of people do that, but that's not what I'm doing. No, so when a plant-eating insect tries to find a plant that it wants to eat, particularly the ones that we work with, we call them specialists because they only have one host plant, really, the weed. It typically flies around first, and during that, before it lands, flying phase, it sees and smells plants and does so much better than we do, much better, particularly smelling a plant. Every plant has a very specific scent, but it also sees it. Of course, insects see completely different than we do because they have these complex eyes and they see ultraviolet and infrared light sometimes. So completely different vision. But that we can measure. We can measure the scent of a plant, and we do. And we can measure what we assume the insect sees. We can even see what exactly an insect gets excited about, because we basically do, like when you go to your doctor and take a neurological exam, we can measure if there's a certain molecule the insect really likes, or if there's a certain wavelength of light an insect really likes. Yeah, and that's basically what we are doing. So it's I didn't show you all the machines that are involved, but there's a good fair amount of machines involved.
Thanks.
Thanks, Mark. So I think we'll move on to Harriet Hinz with CABI Switzerland. And again, if there's any additional questions for Mark, we'll leave time at the end of all the presentations. So thank you again for joining us, Harriet, and go ahead and take it away.
Okay, you can see my screen, okay?
Yep.
Okay, perfect. So as John already said, CABI is an international, intergovernmental, not-for-profit organization. We have our headquarters in the UK, and we have 12 overseas centers, one of which is in Switzerland. So I'm the director of the center, and I'm also leading the weed biocontrol section. We are quite lucky, apart from lab, office space and a quarantine, we have a huge garden space so we can conduct experiments right at our doorstep. We currently have 18 projects for North America, weed biocontrol projects. 8 of these are co-financed by Montana, mostly the Noxious Weed Trust Fund. We mostly work with insects and mites, but we also have fungal pathogen expertise through our UK center. And most of these projects are financed by a so-called consortium approach, which means that several entities with the same weed problem get together, put money in a pot, and then we do the research. That's our team. We have, there's 5 project scientists, 2 garden technicians, a lab technician, some temporary students, but we also rely on a large network of external collaborators in order to conduct open field tests, to facilitate field surveys. That's our funding sources, about 15. The 5 largest ones would be USDA APHIS, Army Corps of Engineers, the states of Montana, Wyoming, and British Columbia. Most of it comes from North America. In 2020, that amounted to $910,000 US, which sounds like a huge amount of money, but when you divide that by 18, we arrive at about 50,000 per project. So it's much more, medium-sized then. In the following, I'm going to talk about the 4 red highlighted projects. So let's start right away with hoary cress. That's a project that started in 2001. We currently have 5 different entities that fund that project, and Mark Schwarzländer, who just talked, is the consortium chair. Before I talk about our work, I would like to talk about an aerophyte mite that Jeff Littlefield at Montana State University investigated and petitioned for release. As you might know, mites are extremely small. On the left-hand picture, you see a single mite on a needle tip. But they can do quite some damage. On the left-hand side, you see that this mite attack hugely reduces seed production of the plant, and occasionally it can also attack secondary shoots and stunt the growth of plants. It's also extremely specific. So Jeff received the permit to release the mite in 2018. In 2019, he started releases at 2 sites in Montana, and he was able to recover galls at both sites in 2020. So that's already a very good start. The mite established, it overwintered successfully. But as you can imagine, a lot more, a number of years will be necessary so that the insect really reaches some higher densities, and that Jeff is then able to redistribute it across Montana and other states. At CABI, we are working with a gall-forming weevil since 2003, Pseudorchestes cadariae, which attacks the shoots and can kill shoots, as you see on the left-hand picture. It attacks bolting plants and rosettes of hoary cress. We first petitioned it in 2011, but APHIS and TAG requested additional tests that we did. This insect has a relatively broad physiological host range. So the host range that we determine with very artificial no-choice tests, but it does have a narrow realized host range. So that would be the host range under natural field conditions. We re-petitioned, or we resubmitted a petition in collaboration with Mark at the beginning of last year. TAG's response was a little bit ambiguous. They didn't really deny they didn't really say you cannot release, but they didn't really recommend the release either. In the meantime, we had a discussion with USDA, APHIS, and TAG, and I think we did find a way forward, and we are currently planning additional tests. Then we have a second weevil, a seed feeder, which is very specific. So specificity is not the problem. It can destroy up to 84% of the seeds. You might wonder, okay, why do we need another seed feeder if the aerophyte mite is already reducing seed output. Well, we don't know whether the mite will establish at all sites. Will it reach really high enough densities? It's always good to have another insect up your sleeve. Just quickly, the pictures, that's the adult, an egg, seed.
Meeting.
And then you see a larvae mining in the seed, destroying the seed, but also adult feeding on the right-hand picture can destroy seeds. We think that we will likely submit a petition in about 2 years. I now switch to Russian knapweed. That project is even older. It started in 1997. So these projects really go on for a long time. You have to imagine that a lot of these insects are univoltine. They only lay eggs once a year. So we cannot work with them usually during the whole, during 12 months a year. Again, we have several funding entities, Montana, USDA, and Wyoming. Again, I will start with the insects that have already been released. In 2008 and 2009, 2 gall formers were released based on work that we did in Switzerland. Wasp and a gall midge. The gall wasp, we just received actually an email by Dan Bean last November, and he was very excited because at one of his long-term monitoring sites in Colorado, he found knapweed plants clearly stunted and covered with galls. So this is where the picture on the left comes from. He actually found plants up to 100 galls per plant, and they made an additional 1,000 releases across Colorado. In California, I read that they had a steady increase in the number of galls from 25 in 2017 to over 4,000 galls per square meter 2 years later. So from 25 to nearly 4,000, that's quite something. They did already find a seed reduction by about 70%, but it really takes several more years of long-term monitoring to really quantify what these gall formers are doing, under which conditions, is it at all sites, do they like moisture sites, etc. It's only with this kind of research that you can then really recommend to people how to use the insects and also manage expectations. My colleague Phil Weil is working with a third potential agent, a leaf mining weevil that he collected in Kazakhstan. And in the beginning, we thought, wow, it's a leaf mining weevil. Should we even start working with it? It's a little leaf mining. What can it do? But it's really the sheer number of insects which have an incredibly short generation time of only 3 weeks at 25 degrees that can really damage the plant, as you see on the lower picture. Because this insect is reproducing so fast and he can actually have a more or less continuous rearing in quarantine, he was already able to test 48 different test plants. Only 3 supported some limited adult development. Now, in order to really show that the insect is environmentally safe to be released, in the ideal world, we would like to conduct an open field test in the area of origin where the insect comes from in Kazakhstan. But as you can imagine, due to COVID restrictions, we were not able to go last year, and it's still unclear to Phil whether he can go this year. So that's it for Russian knapweed. I continue with oxeye daisy. That project started in 2008, and my colleague Sonja Stutz is responsible for that. Again, we have several different funding agencies, and we have 3 consortium chairs, Alec and Rose in Canada, and Jeff Littlefield for the US. Sonja is working with 3 different insects. The furthest advanced one is a root mining moth in the genus Dichrorampha. She completed tests with 69 species. It was able to develop in 11, again, under no choice conditions, so under these very conservative test conditions, but in the open field, so much more natural conditions, only 2 ornamental species were accepted. Now, one of them is just a daisy, and as you might have the plant even in your garden. It is a very common and valued ornamental. So Sonia had to make sure that at least there is no impact on the plant, and she was able to show that there's no impact on biomass, no impact on the number of flower heads. So the ornamental value of the plant is not is not concerned, but the insect does have an impact on oxeye daisy. So a petition is really about to be submitted in the next couple of weeks for the US and Canada, for actually both to both countries. Then there's a second Dichrorampha species, a stem mining moth. She collected in Norway. And this one is a little bit harder to work with. So far she was not really able to establish a good rearing colony. And that's also one point I would like to make. It's not as if we collect the insects and we can start working with them. Sometimes we really have to start from scratch, find out, okay, where are they actually developing? This one was recorded in the literature as a stem miner. But Sonia actually found that the young larvae, they roll the leaves together and mine in the leaves. So it's all these kind of small things that you have to investigate before you can even start conducting meaningful host specificity tests. And this is what she will continue doing. So this one is right at the beginning. And then there's a third species, a root-galling fly, collected in the Czech Republic, that reduces biomass and the number of flower heads. It does look quite specific. There was some gall development on non-target species, but quite inconsistent. And these kind of potato type things you see attached to the roots of oxeye daisy on the left, that's the galls of this fly. So all of these species have potential, we think, but need more years of testing and investigation. And I come to the last project, common tansy, started in 2006, again, several funding agencies, same consortium chairs. Again, Sonia is working with 3 different insects. The first one is a stem mining weevil. They were able to show that it reduces the stem height. All the host specificity tests are conducted in St. Petersburg, Russia, by our collaborators there, because that's where the insect comes from. And we cannot, it doesn't really lay eggs too well in our quarantine. Of 50 species tested, they found some occasional attack on native Tenacetium species and on a medicinal herb in the same genus, but to a much lower degree than on common tansy. The second one is a leaf-feeding beetle, also from the Czech Republic. Again, this time under no-choice tests, it was able to complete development on some native Tenacetium species, less so under multiple-choice cage conditions. And the next step would be to conduct an open field test in the Czech Republic. And the last insect on common tansy is a flowerhead and stem mining moth with a quite complicated life cycle. Females oviposit in the flowers, but the larvae then drop down, overwinter in the root, and then attack the developing shoots in spring and severely stunt them. Again, not surprisingly, there was some issues with the native Tenacetium species, this time because of their difference in flowering phenology. They flower quite a bit earlier than common tansy. So for Sonia, it's difficult to synchronize them in order to conduct host specificity tests. But in the one open field test that worked out, she only found larvae on common tansy. So again, we have a lot of hope for these 3 insects on common tansy. And that's all I had. Let me know in the chat whether you have any questions or orally. Thank you.
Thank you, Harriet. There was one question in the chat, and I apologize, Ethan. I didn't see your question until we were close to the end, so I'm not sure which insect or which system you were talking about. But if you want to ask your question about galls, go ahead.
Yeah, it would be good if I would know which system the gall, because there were an awful lot of gall formers. So
And the Russian knapweed.
Knapweed, yeah.
Oh, Russian knapweed.
I'm not aware that they can kill plants. No, it's kind of more stunting.
Okay. All right, so I think we're running 10 minutes to the hour. We had taken some questions in the middle. So I'm thinking that as we switch gears to Melissa Maggio and the Montana Biocontrol Project, she'll probably go over the 11 o'clock mark there. But this is a little bit of a switch from all of the research information that we have been listening to so far. The Montana Biocontrol Project and Melissa Maggio really coordinate the movement and monitoring and management of a lot of these biocontrols once they've gone through this process that's been discussed so far. And they're at populations within Montana that we can move around and implement their use as part of an integrated management system. So go ahead and take it away, Melissa.
Okay. So thanks for having me today. And I am going to apologize because I am going to stop sharing my video. I've had some connectivity issues this week. And so in an effort to be proactive, if I stop my video, hopefully that will not happen again. So I'm going.
Melissa, I will say that you're not quite sharing your presentation. You're sharing your presentation and your notes.
Interesting. Now are you just, oh really? That worked. Okay. It's the opposite as it usually is. So technology issues continue. Okay. So thanks for having me. As Bryce said, my name is Melissa Maggio and I'm the coordinator of the Montana Biocontrol Coordination Project. So I'm going to give you just a quick overview of what that program entails and then speak to you a little bit about mostly the established and impactful biocontrol systems that we work with here in Montana. So just as a bit of background, the Montana Biocontrol Coordination Project is a soft-funded grassroots effort that was initiated in 2013 by federal, state, county, nonprofit, and private land managers throughout Montana who saw a need for increased coordination within the state's weed biocontrol program. Because we are soft funded, we strive to provide the deliverables that our stakeholders are interested in. And so therefore we conduct a program assessment every 5 years to ensure that we are doing that. The mission is to provide the leadership, coordination, and education that's necessary to enable land managers across Montana to successfully incorporate biological weed control into their noxious weed management programs. So our most recent assessment was conducted in 2019, and the respondents to that survey indicated that they were most interested in the project focusing on insect distribution, education, and monitoring. So that is where our current main focus areas lie. And this could change after the next assessment that we conduct. But currently, we put a lot of effort into these 3 areas. So for insect collection and distribution, this is really focused on those abundant and effective biocontrol systems that we have here in Montana that I'll give you a lot more information about in just a moment. And we work with land managers throughout Montana. We host a lot of collection days. Last year it was over 40 days that we were out in the field collecting insects where land managers were invited to join us. And if land managers are unable to make it to those collection days, we do our best to collect insects for them and ship those insects to them. We also focus a lot on education, coordinating workshops, giving presentations, developing educational materials, and a lot of those workshops, field days, presentations, we provide a lot of research updates. So what John, Harriet, and Mark provided to you today, I attend a lot of these kinds of presentations, gather information from them so I can share them with folks in Montana. That was another point that the respondents very clearly made, is that they were really interested in being kept up to date on what was going on with biocontrol development. So we really make sure we're providing that information. And then lastly, we put a lot of effort towards monitoring, and most of this goes towards new biocontrol systems. They're our priority, but we also work with agencies and organizations to develop more comprehensive monitoring plans. And so a lot of times within those areas, we're focusing on some of these older, more established systems. And then also additionally, if there's any kind of monitoring need related to research, we also make that a priority. So for the rest of the presentation today, I'm really going to focus on insect distribution because it's for those abundant systems. And then I was going to share just a little bit of information on 2 monitoring projects that we have that are related to a research need. So I guess before I hop into that though, I just wanted to mention some of these other systems that I'm not going to focus on. They're systems that are maybe for biocontrol agents that are less abundant, or weed systems that are less abundant, or new biocontrol systems, but I just wanted to make sure that you knew that these were out there. So Harriet mentioned Russian knapweed, and those agents that she mentioned were released are established and relatively abundant in Montana. We definitely have the ability to get them and move them around. It's just that there's not a lot of Russian knapweed in Montana. And so it's not a system that's really in high demand. And so we do work with it, we do move it around. It's just not as common as the ones I'm going to dive into in just a moment. Also diffuse knapweed. I don't think I have ever had a request for biocontrol for diffuse knapweed, but again, that's just because there's not a lot of it in Montana. There are biocontrol agents that I can help folks get if they're in need of that, or if diffuse knapweed starts to increase, we do have biocontrol that we can work with there. Tansy ragwort, there's just a few isolated populations of that here in Montana, but there are biocontrol agents that have been very effective in those isolated areas. So if that starts to increase, or if there's a new population that pops up, we do have the option of utilizing biocontrol to help manage it. And then the last 2 are more in the research phase still, I would kind of say, in the sense that the management of those biocontrol agents is still in the hands of the researchers. So for salt cedar, there is a defoliating beetle that is abundant in some parts of the US, not so much abundant in Montana, but we have researchers at MSU and with the Forest Service that are working on really building up, rearing it in lab to build up populations so that hopefully we can get established populations in Montana. And the reason that we need to do that is because this beetle cannot cross state lines. We can no longer move it from where it's abundant into Montana. But if it is in Montana, we can move it around from there. So currently these researchers are really working on building up those numbers so that we have it as a biocontrol system that we work with, just like these other abundant systems I'm going to tell you about. So if salt cedar is an issue for you, I can't necessarily help you get insects, but I can put you in touch with the researchers who are working on that. They're really making very targeted releases at this point. And Harriet, excuse me, mentioned white top and Dr. Jeff Littlefield at MSU, and that mite that was just recently approved for release. And Jeff is really still managing the areas where it's released. It's still kind of in that initial release research phase. So again, same as with salt cedar, I can't necessarily help you get these insects. We're not collecting them, but I can definitely put you in touch with the people who are managing them at this point. So now I'm going to start speaking to the effective biocontrol systems in Montana. And so these are really yellow toadflax, Dalmatian toadflax, St. John's wort, leafy spurge, and spotted knapweed. These are the systems that we work with most commonly. So for Dalmatian and yellow toadflax, there are two species of stem weevils that attack these toadflaxes. And it's really important to keep in mind that they are species specific. So Macinus janthinus only attacks and impacts yellow toadflax, and Macinus janthiniformis only attacks and impacts Dalmatian toadflax. And this is very important to know because they look very similar, and there are commercial distributors that sell biocontrol agents here in Montana. You can get them from organizations like mine or other school groups. And if you're doing that, I always encourage people to ask questions about which toadflax the weevil was collected off of, because that is the toadflax that you need to release it on. Potentially, you might see some feeding from the yellow toadflax insect on Dalmatian or vice versa, but it's not going to impact that plant and help you manage it unless it's on its specific species. So important to remember that Macinus janthinus is for yellow toadflax and janthiniformis is for Dalmatian. A little bit of on the biology of these species. So they can emerge as early as April and be active through July. This is pretty weather dependent. Here in Montana, typically we see the yellow toadflax weevil emerge probably mid-May, and we continue to collect it till probably mid-June. And for Dalmatian toadflax, that weevil is more like the month of June. So the yellow toadflax insect emerges and is collectible a bit earlier than the Dalmatian insect. The adults feed on the young shoots, leaves, and stem tips, excuse me. They overwinter in the dead toadflax shoots. And as with all of the biocontrol agents we have pretty much here in Montana, there's just one generation per year. That's really important to keep in mind because those adults that you release, you need them to be able to find each other, mate, and lay eggs so that you have insects the following year. If you're wondering if you already have these insects in your weed infestation, you can go out during that time that I told you those adults are out. So probably beginning of June is a good time for both of them, and visually inspect to look for the weevils, or you can sweep net. I do discourage sweep netting if it's all possible for people to just go out and visually inspect, because those sweep nets can snap the stems, and if there's already been eggs laid on those stems, they will not complete development and be adults the following year. We often take trays and tap the plants onto the tray and the insects will fall off. So that's another good way to look for them. You can also dissect stems in the summer and look for larval feeding, larvae or pupa. And from the fall to the following spring, so through the winter, you can go out and look for those old dead toadflax stems and dissect those, and you'll find adults within. So here's an image of a Dalmatian toadflax stem that has been very impacted by the stem weevils. You can see all the little feeding holes there and some discoloration as well. And I guess I should show you up here on the left-hand side, that's yellow toadflax that's being impacted by these weevils. You can see the tips of it are yellowed and kind of spindly. So next I was going to talk about St. John's wort. St. John's wort has not been something that has really been in high demand the last 10 or probably more than that years, but probably in the last 3 years, I've really heard some increasing concerns about St. John's wort, and I don't know at the moment if that's because St. John's wort is increasing or if it's all just part of the cycle that we can commonly see with St. John's wort and its biocontrol system. But there's some very capable researchers who are looking into it to see if something more is going on beyond just like a downward trend of biocontrol agents and us just needing to make additional releases. So there are a number of, or a few agents that you might see on your St. John's wort. The one that we really focus on collecting and releasing is chrysolina. There's also an inchworm and a root borer that you could potentially find, and sometimes we do move those around, but again, most of the emphasis is on moving chrysolina, or it's also commonly known as Klamath weed beetle. So a little bit on the biology. The larvae emerges in early spring and it feeds, and then it goes back to the soil and pupates and emerges as an adult, and the adults come up and begin feeding. This is probably right around the time that the flowers are starting to open on St. John's wort. And then oftentimes those adults will go back to the soil and take a rest during the hottest, driest months that we have. And then they'll reemerge in the fall, feed a little bit more, and then start laying eggs. So majority of the damage is done by larval feeding, but there can also be really significant damage by the adults. You can see in the picture here that they really do congregate, and it's a defoliator. So the damage is pretty striking, and those adults are pretty showy. They're probably the size of a pencil eraser, and they're metallic and shiny, very pretty. So people really notice them. If you're wondering if you have these insects, the best time of year to go out and look is just as those flowers are starting to open, and you can visually inspect for those adults. They're pretty obvious. You can take a sweep net out. That's another way to do it. Also, the plants will appear to be stripped and wilty, and that, you know, was probably from a combination of both the larval and the adult feeding. You can see this is a picture of some of the adults in my hands, so you kind of have some scale there. Next, I was going to speak a bit about some of the leafy spurge biocontrol agents. So there's a number of biocontrol agents that have been released to help us manage leafy spurge. We really focus our efforts on collecting and redistributing the flea beetles and the stem boring beetle, so Aphthona and Oberea. There are 6 species of flea beetles that have been released. There's 2 species that have really established and are thought to be the most impactful here in Montana. Those are Aphthona nigroscutis and Aphthona lacertosa, more commonly referred to as the brown and the black flea beetles. They emerge in May and continue to be active till probably late July. Again, that's very weather dependent. Here in Montana, I would say the prime active time for the flea beetles is early July. So those adults, they feed on the leaves and flowers, and when they're in really high numbers, they can really strip the plants, and the feeding damage is pretty obvious. The larvae feed on the fine root hairs, and that's where majority of the damage that these flea beetles inflict is caused from, is feeding on the root hairs. Oberea is a stem boring beetle. So the pictures on the bottom, the little shiny brown beetle is a flea beetle, and the picture next to it is Oberea, or the stem boring beetle. It's oblong, black with a red head and really long antenna. It's thought to not inflict as much damage as Aphthona, but they're really great in combination with one another. Oberea does a good job of thinning out a stand, and that's where the flea beetles tend to prefer. They like it where the sunlight can penetrate to the soil, and Oberea kind of thins those stands to that point. So the adults feed on the leaves and flowers, and the larvae feed on the stem pith, and they tunnel their way down to kind of the top of that root system. So another thought that or something else that I've heard people discuss is that potentially Oberea might be a good option for areas that seasonally flood. So when the soil is saturated every spring, those flea beetles have a really hard time. They just get drowned out basically. And so this Oberea is at the base of the root. So they might be able to survive in these areas that seasonally flood much better than the flea beetles can. They also have a much slower population growth than the flea beetles. So it takes a while for them to build up their populations, but we've seen some really significant populations of Oberea in the Missoula area and also kind of over in Jefferson County as well. If you're wondering if you have these agents for the flea beetles, you can look for craters, which I will show you a picture of in just a moment. It's basically just a dead area where those flea beetles have kind of congregated and killed all the leafy spurge. You can also go out and sweep net for them. And again, probably prime time would be early July. And I would definitely suggest not trying to visually inspect. They're definitely not easy to see just walking through an infestation. There's been a number of times where I've walked through a field and thought that there weren't very many flea beetles, but I grabbed a sweep net anyways, and there were quite a few. For Oberea, you can look for egg laying damage, but I would suggest taking a sweep net out. That's probably the best way to look to see if you have any of them. And they're out a bit earlier than the flea beetles. If flea beetles are most abundant early July, Oberea is probably most abundant 2 weeks before that. So I just wanted to show you a series of pictures. This is something that we commonly see with leafy spurge biocontrol and also with St. John's wort biocontrol. I kind of hinted at this, but I didn't get into it. There's a very cyclical nature with the weed species and biocontrol for these weeds that have really extensive root systems. So at this site, there were 50 flea beetles released in 1989, and this picture was taken in 93. And now when we move flea beetles around, we're putting them in containers of 1,000 to 2,000. And in 89, they only released 50, so significantly less than what we move around these days. So in 93, they saw the first sign that the flea beetles were establishing and impacting. And what they saw was this crater, that area of dead vegetation in the middle of that picture there. That's probably where they made their initial release. And those flea beetles really built up their numbers, killed everything off there, and then they start spreading out throughout that infestation. So this is what it looked like in 94. You can see that there is still leafy spurge here. Up at the top, it's still flowering. Down lower in the picture, it's not really flowering, which is something we commonly see with biocontrol. You can see that the bunch grasses are coming back, which is great. 95, you know, the spurge is more visible. You see it flowering, but it's very sparse compared to what it was in 93. It's single stalks rather than a monoculture. You see a lot of good bunch grasses. 96, not much spurge. Looks like cheatgrass is moving in though, unfortunately. 97, starting to come back. The spurge is making a comeback. And then 98, it's nowhere near as bad as it was in 93, but there's a bit more. So this is really common, like I said, for these weed species with really extensive root systems. Usually the weed population is at a high point when you release your biocontrol agent. That biocontrol agent increases its population to the point where it's suppressing the weed species. But because these are species-specific agents that we're working with, once they reduce that weed species to a point, they have to disperse to look for food and habitat to overwinter in. So oftentimes a number of the individuals leave, some still remain in the system. But unfortunately, these weeds with these extensive root systems are able to make a comeback. So then their population increases, and then that, you know, triggers the insect population to increase. And so you see this cycle over and over and over again. But even at the high point, after the establishment, there's significantly less than prior to establishment of biocontrol agents. I can tell you for the bison range, that when they first started releasing chrysolina for St. John's wort, they estimated that they had about 10,000 acres of St. John's wort. They released those biocontrol agents, I think, in the 1950s. And now on a bad St. John's wort year, they have about 1,000 acres of St. John's wort. And on a good year, where there's not much St. John's wort, they have about an acre. So there's a cycle that still occurs there, but even on those bad years, it's significantly less infested acres than prior to the establishment of biocontrol. So next, spotted knapweed. I think we've released about 13 agents here in Montana for spotted knapweed. But the ones that we work most with, or that we consider to be the most effective, are the root weevil, Siphochleonis acades. There's 2 flower head seed feeding agents. One's a weevil and one's a fly, excuse me. And then there's also a root feeding moth, which is Agapita. So I'll tell you a bit about these agents. So Siphochleonis acades probably gets the credit for being the most successful biocontrol agent for spotted knapweed, but I really think it's because of the combination of all of the agents. Like I said, we have a number of agents that attack different parts of the plant, and I think that's where the success really comes from. But most people know about this root weevil. It inflicts most of its damage in the larval stage, where it's feeding on the inside of the tap roots. And this is how it, excuse me, overwinters. The adults emerge and feed on the leaves and flowers and stems, and they mate and lay eggs. Again, just 1 generation per season. These adults are out and active probably the month of August here in Montana. Agapita is a little yellow moth. It also overwinters as a larvae in the root. So oftentimes you can find both of these larvae in spotted knapweed tap roots. They emerge a little bit earlier, or they're out and really in their prime a little bit earlier than Siphochleonis, probably even in June is when we see them most abundantly. So if you're wondering if you have them, you can pull up spotted knapweed and dissect roots pretty much anytime, looking for larvae. The root weevil tends to be more of a C-shaped larvae, where the moth tends to be more of a slender, straight larvae. You can also sweep net or visually inspect. Agapita is a very delicate moth, and so and it's also a very good flyer. So we do not put any effort into collecting and moving this. If we did, we would probably have a lot of mortality, and so it's just not worth it because it is such a good flyer. It's in most infestations of knapweed that I visit in the summer months, but Siphochleonis does not fly, and we do therefore put some effort into going out and collecting and distributing these insects and moving them around. So for the seed head feeders, there's 2 species of Laurinus and there's 2 species of Urophora. Laurinus is a weevil, Urophora is the fly, and these attack the seeds. So these are not considered to be plant killers, but they're definitely an important biocontrol agent. Reducing spotted knapweed seed is also very important. There's been some studies done that indicate, because of the establishment of these 2 agents, that they can reduce the amount of viable seed that knapweed produces by 97%. So that's very significant. So for the weevils, the Laurinus, the adults will feed on the leaves, and then they lay eggs on the flower heads. The larvae feeds on the seeds. They hatch into adults, and then they emerge from those flower heads and overwinter in the soil or the ground litter. For the flies, the adults lay eggs in the seed heads, and the larvae tunnel to the base of the seed head, and they overwinter as larvae in the seed head. So if you're wondering if you have these, a good way to look for them is to check out your seed heads right now at this moment even. So the picture in the upper corner there, that is a blowhole, basically. That is where the Laurinus emerges before it overwinters in the soil. And these are really obvious. You'll see these in your old seed heads, and it's a good indication that you have Laurinus already established in your spotted knapweed infestation. If you're looking for Urophora, same thing, go out, grab some of those old seed heads right now, dissect them, and look for the larvae. It looks like a little grain of rice, basically, at the base of that seed head. So we do not put much or any effort into collecting and moving these species. They're very good flyers. There's very few infestations of knapweed I've ever visited that I don't find evidence of these species already established. Sometimes we do move Larinus around, but it's really targeted. If it's a new isolated infestation of spotted knapweed that maybe Larinus hasn't made it to. Also in really high elevation or harsh climate environments, we'll sometimes move Larinus, but those are mainly the only times that we move them. So now I'm going to kind of switch gears and not necessarily talk about abundant or established biocontrol systems, but talk about some projects that kind of have more of a research focus. So the first is for Canada thistle, and this is focused on the Canada thistle rust, Puccinia punctiformis. This is a rust that is host specific to Canada thistle, so it only impacts Canada thistle. It's also naturalized, so it likely came over with Canada thistle. There's back into the late 1800s, there was even indication that folks thought that this could potentially be a good biocontrol agent, but pathogens are just notoriously difficult to work with. And so it took a long time to really figure out how to do that. ARS picked up some work, I think in the 90s, and they, found a lot of good information through research, and then Colorado Department of Ag picked that up, I think around 2010, and their intention was to develop a landowner-friendly protocol to move this pathogen around just like we move insects around. And so they began sharing the rust with other Western states, I think in 2017. That's when we received it anyways. And so we began inoculating sites in the fall of 2017. I think we had about 17 sites that we were working with from 2017 to 2019. And then in 2019, we discovered 2 of our inoculated sites that had really significant infestations to the point that we were able to harvest our own infected material and create our own inoculum, basically. So we ground that up, and we had enough inoculum to put out at a number of sites in Montana in 2020. So we monitor these sites in the spring and we inoculate in the fall. And so the spring of 2020 was kind of challenging for us with all of the COVID travel restrictions, but we wanted to make sure that we monitored every site that we put this inoculum out at, just so we had good information on, what it was doing, what sites it was really establishing at, and maybe what sites it wasn't. So we were able to work with partners and get to a number of sites, maybe a few less than we had intended, but by the end of 2020, we were able to monitor and inoculate 72 sites throughout Montana. The pictures on the sidebar here are from one of the sites that we harvested from in 2019. The top picture was taken when it was inoculated in the fall of 2018, and the bottom picture was taken last fall. So you can see that there was not much Canada thistle left by last fall, so we were no longer able to harvest from the site. There was a near eradication from Canada thistle there. But we did still have one harvestable site last fall, and we were able to harvest enough material to re-inoculate our 72 sites this fall if they need to be re-inoculated. I'm also excited because we're working in partnership with Drs. Seipel and Rue at MSU to utilize all this data that we're collecting to develop predictive tools. So the intention is to develop some sort of tool that helps land managers decide if the rust is an appropriate tool for them, if it's going to work at their site or not. This is the distribution of our Canada thistle rust sites throughout Montana. And then the last project I wanted to tell you about is, kind of goes hand in hand with what Mark was talking about. We are looking at non-target impacts of the hound's tongue root weevil. So there was a science advisory panel that was organized by the Montana Invasive Species Council in the spring of 2019, and they brought together a number of experts in the field, whether it was about the weevil, the plant, or regulation. And the intention was to scope the potential for approval of the hound's tongue root weevil as a biocontrol agent in the US. A number of recommendations came out of that panel, but we utilized 2 specific recommendations to look for non-target impacts and to develop tools to help land managers identify the weevil, to develop a protocol to develop a proposal that we submitted to USDA BCHIP and received funding for in early 2020. So our project is modeled after a post-release research project that was conducted in Canada. And our methods are pretty similar to this project, with the exception of making weevil releases. As Mark mentioned, it's illegal to move these insects, even if you find them in your backyard. So we worked with Drs. Mangold, Singh, and de Klerk-Float to develop our protocols for this monitoring. And basically, we find sites where there's hound's tongue, desired non-target Boraginaceae, and the weevil all present within our 5 by 5 meter plot. And we collect data on all the plant species, the hound's tongue, and the desired non-target for 3 consecutive years. And at the end of the 3rd year, we'll destructively sample a subset of the hound's tongue and non-target Boraginaceae species to look for egg and larvae. So in 2020, again, we had some setbacks with COVID-related travel restrictions, but we were able to identify 9 sites in Montana that had all those things we were looking for, and we established plots at 8 of those sites. The 9th we'll add this year. We did additionally search for sites in Washington and Idaho in the fall when we were allowed to travel out of state, but we did not find any ideal sites. So this year in 2021, we need to add 2 additional sites in Montana, 5 in Washington, and 5 in Idaho. This graphic shows you the distribution of the hound's tongue root weevil in Montana that we've found so far. This definitely isn't the end result. It's probably in a number of other counties, but green is where we've confirmed the presence of the weevil. Turquoise is where we suspect it is. Saw some suspicious feeding damage, but we're not able to find any eggs, larvae, or adults. And the stars are where the monitoring sites are located. So that is everything, and there's my contact information. So feel free to reach out if you have any questions.
All right, thank you, Melissa. We have covered a ton of information today. And I think I want to thank the speakers, and I want to thank everyone for sticking with us through all of that information. And I think we definitely have some time for questions if people have them. I'm not seeing anything in the chat, but.
Hey, Bryce.
Yeah, John.
I did want to point out for everyone who isn't familiar that the service that Melissa does is fantastic for the state, and it's free.
Yes. Yeah, and I think, Jamie, I'll kind of kick it to you for some closing comments if you have any, but I think we'll certainly make available all of the contact information for the speakers today if you want additional information. And then I believe the webinar here was recorded, and I think we'll also work with the speakers to make sure that their presentations are available through the MOA website. There is one question about Melissa's contact information, and she just answered it. So nice job.
Yeah. Wow, thanks everyone. This was fantastic. I appreciate your time and your efforts and the outreach. We have a variety of guests today, so it's always good to be able to get this information to new places. So yes, we will post this on our YouTube channel, and then we'll also make the slides available on our website. Thanks everyone for attending. Thanks speakers for all your wonderful presentations. And if there's a need, or if anyone wants to do another presentation in any way, I'm happy to host and pull those things together. I think it's important to get this out. So thank you.
All right.
Thanks, Bryce.
Yeah, thank you, Jamie. Thanks,
All right.
everybody.
Bye-bye.
Enjoy the sunshine.
Guys.
Thanks for organizing.